Well, good evening, everyone. I'm Paul Saraja, a cardiologist at the Minneapolis Heart Institute and the Minneapolis Heart Institute Foundation in Minneapolis. And I have the distinct pleasure of welcoming you to the part 3 of the Imager and the Interventionalist. It's a webinar series that our foundation, Cardiovascular Innovations Foundation, is sponsoring, featuring interventional imagers who are experts in their field. Tonight, I had the distinct pleasure of welcoming Dr. Didi Wang from Detroit, Michigan, and we're going to get to know her up close and personal here in a moment. But for those of you who are interested in claiming CME, these are learning objectives. This activity is accredited for 1 credit, which you can claim at our website if you choose to do so. We'd like to thank our sponsors, Abbott, Boston Scientific, and Medtronic for helping support this series, which we feel is very important to the education of the public. And here's a link for where you can claim that CME credit. Our next webinar, live webinar, will be the approach to patients with polyvascular disease. This will be on Thursday, May 5th, 2022. And for those of you who are available to come, I encourage you all to come and participate in our annual meeting, CVI 2022, which will be at the Hyatt Regency in Denver, July 21st to 23rd. And it's going to be an exciting in-person meeting, and I hope to see many of you there. For those of you who may be interested in travel grants, abstracts, and research support, please go to our website. We have adequate funding to support your needs. For today's webcast, as well as others that have been archived, you can find them at our website, cvinnovations.org. So with that, I'd like to welcome Didi to tonight's feature. And Didi, welcome. It's great to see you.
Pleasure to be here, Paul. Thank you for the invite. This is pretty exciting. I'm actually kind of nervous. So I have no idea what you're going to ask me.
Well, as I mentioned at the introduction, this is really a feature of people like yourselves, who have just really led the field for interventional echo and interventional procedures that require a lot of imaging. And over the past 10 to 15 years, it's been amazing to see the change in procedures and incredibly reliant we become on people like you. And we can't do what we do without you. That's for sure.
Thank you. Thank you so much. I gotta say, we can't have a job without people like you supporting us. I mean, what was it, 3-4 years ago that we wrote our manuscript for interventional imaging physicians and what the training background needs to be. And at that time, I'm like, Joel, do you think we can get Paul to sign on to this manuscript? And not only did you sign on, but you support it wholeheartedly and made it more assertive. And you're like, you guys got to be more assertive in what you say and say that you actually need more important training background and consensus. We're like, well, if Paul says that, then we're putting it in there. So thank you.
Well, I think first of all, I thoroughly enjoyed working on that because I learned a lot from that, as I always learn a lot from you all. I mean, I grew up level 2 trained in transthoracic. I never learned or never held a TE probe. I never spent time in CT and MRI. And the things that I learned from you and Joao and many others, it's really amazing. So I want to first start, kind of just introducing you to people who have tuned in, and I want to get to like where you started, how you ended up where you are, and kind of what your outlook is for the field of interventional imaging. So Didi, why don't you just tell us a little bit about how you started off in this area?
Well, for some of the people out there who know me, I actually wanted to become an interventional cardiologist. So my dream in high school, college, medical school was to be somebody like Cindy Grimes. And I wanted to be the STEMI rescuer. And I wanted to be the, at that stage in time, ER was the hit medical drama. And you had to be wearing George Clooney be wearing his white coat and running around the hospital and being all amazing. And kind of wanted to be kind of like the rescuer. But then when I got to the cath lab and fellowship, That was when statins were there, and we were engaging bypass grafts, and structurally actually hadn't developed yet, and people were doing peripherals. I just didn't see myself doing peripherals in the long run. But then I wasn't smart enough for EP. EP, you have to be super, super smart, and they're super, super patient. Heart failure, it just wasn't my cup of tea, probably. I love the shock portion of heart failure, and that was satisfying. General cardiology, I wanted to do something more, and I wanted to have a niche, but I had no clue what I wanted to do. So imaging by default was the only other fellowship I could do, but I kind of fell asleep in the echo lab, and it wasn't really that exciting for me. But luckily, when I was in fellowship at Henry Ford, Dr. O'Neill was recruited to Henry Ford to start the structural program. At that time, Myra was there and Adam Greenbaum was there too. And for the first 25 TAVRs, everybody filled the room and everybody was in there. But as a non-ACGME imaging fellow, after the first 25 TAVRs, none of the physicians wanted to go in anymore because it wasn't reimbursed. So luckily, the non-ACGME fellow had to be thrown in there to hold a TE probe. That would have been me. And in doing that, I learned the complications they were having. I loved being in the cath lab again. And I had this thrill of just seeing successes with all this new innovation. And then since I was getting trained in cardiac CT and MRI, I was able to really build a great relationship with the cardiac radiologist in our teams. And so I'm like, maybe we should be doing vessel sizing a little bit differently. Here's what we're learning about these devices and the sheets. Is it okay if we modify this? And they were really great in letting me just collaborate and start pushing the boundaries for disruption. And from there, it was just kind of like, what's the next problem that I hear about in the cath lab, and what can we do to change it? And it's kept on growing.
Wow.
So at the end of my fellowship training, I was kind of like, do you have a job for me? Could I possibly stay? I kind of really like this. And I don't see myself going into general cardiology, or because most of the jobs that you see for quote advanced cardiac imaging specialists are a general cardiologist with some bonus MRI or CT reading. I'm like, I kind of like the structural stuff, and I really want to grow with it. And Henry Ford and Dr. O'Neill and Dr. Kim made it happen. So I was lucky. I was in the right place at the right time.
Well, so I want to piggyback on that just a little bit more. So did Bill do the first TAVR in the United States? Were you there?
I was not there. He did that at Beaumont Hospital, but I wasn't there. I don't know what, no, I wasn't. I was in high school.
Yeah, it wasn't, I know it was, I'm pretty sure I know what year it was, but I'm not 100% sure. But I know he was involved from the early beginning. And
yes.
So back then, those first 25 cases or so that you saw, I mean, what was it like? Tell me about the experience of doing it with Bill. I mean, Bill's obviously someone I look up to enormously for interventional structural procedures. What was it like to be in the lab with him?
It was a privilege to squeeze into the room because they started locking the doors because there had too many people going in. And it felt like, you came out and you were in a cloud and a haze. You couldn't believe what just happened. It didn't make sense that all of a sudden, did we just do that? And that patient's coming off the table and they're okay. We didn't do an open chest. It was just kind of very thrilling. And it's a very addictive feeling because that's kind of like what you're doing with all the innovations with TMVRs and all the new technologies. It's a thrill that is very hard to describe because you realize that you're rewriting the medical textbooks and the medical literature. And what's fascinating about it is the cine films and the imaging films, when you put together a presentation and send it to the medical school students or the undergrad students and tell them what we were doing, they get like wide-eyed and bushy-tailed and they're super excited. And you're like, well, you guys want to come in the shadow? It's really, really cool. You can have a career out of it.
Wow. I mean, that's amazing. And so I take it Bill is somebody who keeps his cool pretty well in the lab.
He always keeps his cool. It's very fun to watch him and how he thinks. He always has plan A, plan B, plan C, and the number of devices in his thinking. From when I started in my early career to now, I'm learning from him still how to think about the patient differently. In our general cardiology training, we're kind of siloed. We do echo, we do heart failure, we do EP, and we just transition month to month. But really structural is all about linking everything together from valvular disease to cardiogenic shock. It's no longer a curriculum about one portion of an imaging tool or a catheter. It's really about from plan A to plan B to LVAD, what can you do at each step? And it's a great way to reapproach medicine.
Wow. So I have to, just hearing about your practice and knowing you as well too, I take it that, structural imaging is most of what you do. Is that a fair statement?
It's 100% of what I do. And I do round on the inpatient cardiology telemetry services too, but every patient has a structural heart disease to some extent. So yeah.
So how does, as if I were a early career or trainee who was looking ahead and kind of like you were maybe a number of years back, you're so specialized. I mean, how do you get to focus on just the one thing that you love, that you're passionate about? Because, like what you're saying, a lot of people who are in the structural imaging field, I don't want to say fell into it or have it as an add-on, but they don't get the privilege of doing it 100% of the time like you. And so how does one kind of work into that specialty or become that specialized way in doing things? If you could, how would you advise somebody who's interested in doing that?
Now, that's a great question, Paul. We get that a lot in the field of interventional imaging. Like, how do you protect your time? How do you build a career and have a niche? And it really comes down to partnering with our implanters. And it's not about the individual. The sooner that we understand as a community that the imager is part of our heart team, the better off you will be at quickly scaling a program and growing it. When they look for jobs, it's not about looking for a job as an interventional imaging physician. It's looking for a structural program that's looking to grow. And we're looking to grow in. So I'm fortunately part of a very big structural program, just like you have built too, where you do a lot of different procedures from PVLs to VSDs to devices too. And there's a huge need for imaging physicians to help guide these procedures. One of the most important things is accounting for your time in building these programs is to realize where the programmatic needs. Is the program anticipating and growing the left atrial appendage field, or are they going to be growing in the mitral and tricuspid? And if that's the direction that they want to go, then you know there'll be a need for an imaging partner. And if that's the case, and that's where you guys got to go in and articulate that, hey, this is how we're going to grow, and we can bring these procedures and help these patients much faster if you invest in this time upfront.
So what I'm hearing is it's the passion first.
Yes.
It's the passion, not the program first. Because I think to me, when trainees or people looking for jobs, they're hoping to find what I have or what you have, and it's never out there because
Correct.
whatever we have or you have, somebody else already has, and it's not like it's ready for as a turnkey approach for somebody just to walk in. And what I try to convey, and this is what I'm hearing from you, if I'm paraphrasing, you find some area that you just love to do what you do, and it just organically grows with the team that hopefully shares that same passion with you. Is that a fair way of describing it?
Absolutely. I think you hit it on the head, Paul, when you said it's about the team. It's really about finding the team and what the needs of the team are, identifying it and realizing that that's something that you're passionate about and that you really love. It's not a job. I don't think either you or I will call what we do jobs, because jobs aren't fun. This is actually truly like a privilege to help patients and do R&D and preclinical and kind of disrupt and advance the field. In some ways, it becomes part of just the life that we lead. And we're fortunate to get paid for doing it. And approaching in that mentality allows all of us to kind of remain humble and grow. But absolutely, finding the right team and finding that team that has that kind of hunger that you have will help grow a program without having to feel like you have to go to a program that's already established.
Yeah, and I think that's hard because people. I think at least you and I can show people what works, and hopefully they can go find their way and build it themselves too. So now I learned before we came on that you have an interventional fellowship in imaging that's starting. So obviously teaching others and sharing what you learn with others is obviously a passion to you. I mean, you're doing this because you obviously believe in the future of this. I mean, what do you think about the future, and where will it go? Will there be boards for interventional imaging? I mean, where do you see this field growing?
Well, first of all, I really hope I'm going to be grandmothered into any boards. So I'm done with boards. I don't like boards. But I would say that the future is going to be extremely bright. The job market for interventional imaging physicians is probably the best job market in all of the cardiology graduates. It's incredible. And I just peeked in the participant list, and I have to say I'm very proud that our fellow, who I didn't even tell him that I was going to be on this, Dan Shopsky, is actually on. So I'm going to do a shout out for him on this specifically. Yeah, I think the future is extremely bright. And the beauty about this is we're at the beginning of something really, really cool. We're seeing the early innovation, and the innovation curve is in the early disruption, where you don't have really guidelines for interventional imaging training. Everything's being developed. So every single trainee, every single early career faculty who has any interest in interventional imaging can kind of help shape the future. And never before in medicine, even in structural, do we have that opportunity to actually build this career and curriculum for the future generation. So I think that's really, really exciting.
Well, I think, you mentioned the paper we wrote a few years ago, and that was a large part of it. I felt like, well, I got to be part of this. I got to get in. And it's kind of the way I saw it. You're right. It's so amorphous right now. There's so much room for creativity because there are really no guidelines. There's no.
Yes.
Maybe there'll be board certification someday, but the requirements are actually being set by people like you in the field about the standards. You're writing about the standards, you're describing what needs to be done. It's really great to see. So when I was a fellow, a number of years ago, one of my senior partners told me that, well, it was actually Ted Feldman. I remember this now. Ted was at a meeting, and he was giving a talk to young trainees, and I'm not that much younger than Ted, but young enough. And
younger.
And Ted said you're not a structuralist if you haven't cut a catheter, reshaped a wire, and inserted something into a patient that is off-label.
Or use a hair dryer on the end of it too, and blow dried it from Home Depot. Yeah.
Something like that. And his point was that the structural world is so early, and that there's still so much to develop that we're making it up as we go. And
the incredible part about it too is, not only are we collaborating, but it's also international collaborations around spectrums from R&D engineers. I mean, we went to medical school. Who thought we'd be working with engineers and wishing that we knew C++, and learning how to use all these different technologies, including AI. It's a world of opportunity. And the best part about it is manuscripts. If you think about it, writing, we don't need 5,000 patients, 20, 10, 30. We got a Jack Interventions possible interventional manuscript possibility. So
it is funny how that is true. A case experience of 20 tendines, I'm like the world expert.
Yeah, you are. It was on the cover. I loved it. We cited it.
So now I want to ask you one other thing before we move on to your presentation. So I know you do a lot of 3D printing in your practice, and we try to, and that's a very important part of, I think, being a structural interventionalist, is learning to look at a 2D screen and see the 3D image. As an imager, I think you guys do that naturally because that's what you do. But you also use 3D printing in your practice. Can you tell us a little bit about that and how you use it, when you use it, and what benefits do you see?
Absolutely. So 3D printing is kind of like my dictionary for translation purposes. I learned 3D imaging and 3D TEE by doing 3D printing. And what I found is that, in the imaging world and in the implanter world and on the heart team, every single person part of that heart team has a different type of depth perception and different type of training background. And, for NASA, I've been told that when they screen astronauts for who has three-dimensional perception. I don't think we do that for medical school, or nor do we do that for structural or imaging. So it's hard to say who can see the yellow 3D image and see the depth, and who can see the yellow 3D image and see yellow, and then who sees a 2D image and sees it in 3D, and who sees it just as black and white. So this became a tool to translate. I mean this on my image, but this is what I'm actually talking about. And your catheter is doing this, and if I can't draw it on there, let's just actually put it into the physical model to show it. So we started with the 3D printing actually out of necessity. It wasn't like, oh, we're gonna make a niche out of 3D printing and be known for 3D printing. It was actually when Bill and Myra and Adam had a patient that needed a TMVR. They had severe mitral annulus calcification. And so the first patient they did, we had it was a compassionate case, and we put an early sapien in, and it went in completely fine. There was no pre-procedural planning besides I did some transapical access, so there was one puncture or anything. And like a 26 early sapien went fine. Patient number 2, the valve actually embolized. And then patient number 3, the valve actually obstructed. So we went back and looked at CTs and started like, what can we not see? What don't we know? And we started printing it and looking at, okay, how do we guess how we're not going to obstruct in the future? So our first few 3D prints, we would take this hard plastic model off and just eyeball the gap between the valve and the LVOT. And by our eyeball test, we're like, I think we're nervous. I don't think we're nervous. And because we had no cutoff and we had no idea how to get to it. And then after we accumulated enough of these 3D prints and using the software, that's how we got to the neo LVOT. But all of that in the early TMVR, we were doing it between like what, 2013 to like 2017-ish. It was like 4 years to accumulate almost like 35 patients. So there was no way that we could say there's a value in the 3D printing with 35 patients in over 4 years. Scientifically, it wouldn't hold water because it'd be like, well, takes too long. There's no value in scalability in the real world. So we started doing 3D printing for everyday stuff, left atrial appendage closure. And we used that to start justifying we could do procedures faster, concomitantly with CT. And that's kind of how it took off. We don't need to do that anymore, but the 3D printing still stays with us because we use it for all of our clinical trials. Because the clinical trial teams are made up of different people of different medical or engineering backgrounds who may or may not understand the imaging or the imaging that we do in our style. And we might not understand the catheters from what they understand in their style. So again, we use it for all of our clinical trials and our clinical trial patients or an extremely high risk patient that we just want to make sure we have all the ballot solutions to.
So you use it to communicate to people who may not be able to interpret the images. You use it to test devices. I presume like, you have a new technology and you're not sure that the catheters or the devices fit within that. But now, I gotta think that you can look at an echo and think 3D and know what that 3D print is gonna look like.
It's funny, I can to some extent, but I'm always learning. So in my office, some people joke that I have a creepy office because I've got like 2,000 3D prints in there. I look at the 3D prints and I discover something different on the anatomy every single day. Like, oh, I never considered that angulation. And it's because we have a difficult case. I'm like, oh, so the IVC takeoff and the RA relationship to the fossa actually affects my catheter pivot point. Whereas 5 years ago, I'd be like, oh, it's just a small fossa. And then you need an anterior curve. And nowadays, I'm like, oh, now the left atrium doesn't change the same shape. It actually morphs differently. And that's why I need a different depth for this patient. So little things, depending on what devices are being developed, affect the way that we visualize the 3D prints and how we image. So the education is never ending. We also found as a bonus, it really helps our fellows in their TEE imaging. And it also helps our patients in consenting for procedures because instead of our drawing stuff, they actually can feel it and they have a little bit more comprehension to what we're doing.
Well, that's it. That's how I'm going to get my TE certification. I'm going to have to start 3D printing.
You're always welcome on my side of II beam.
So do you have a 3D printer in house, or how do you do it? Is it contracted out? How does it work?
No, so we actually have 4 3D printers in house. We do all our in-house 3D printing, and how we started is with philanthropic donations. And we had a lot of really kind patients who believed in the 3D printing and the power of this and invested in this. So we formed a grant and foundation. The Ford Foundation donated a lot of their early days money for this until we could justify the clinical workflow and cost efficacy of doing it too. And we don't outsource primarily because we want to be able to control the actual accuracy of the model. And what we want to do is segmentation. Otherwise, we're kind of like teaching other people externally what we need, but not getting the results we want in a real time and fast turnaround too.
So who does the actual printing and analysis? Do you have fellows or trainees or cardiologists, or is it you who does the 3D printing? How is it uploaded and printed?
So we have an amazing structural imaging team composed of CT technologists and coordinators who we train to do 3D printing, who we train to do segmentation, and then we oversee the whole entire process. 3D printers are actually just across from my office. So we have a nice setup that with the ventilation and everything, how to clean, and all of our alcohol buckets. It's a whole real process. It's kind of fun and nice.
Seems like you could also just have a little fun with it and print things besides hearts.
Yeah, well, we have done kidneys, we've done skulls. They're not as cool as the heart, but it gets a nice wow factor if after a while we get bored. People like it a lot.
That's great. That's great. Well, thank you, Dee Dee. So we're going to spend the second part of tonight's webinar on your presentation, and if you'd like to go ahead and share your screen and take us through what you want to share with us as an interventional imager.
Absolutely. So I thought it'd be interesting just to approach surgical valve selection and be a little bit controversial. Paul, you've actually pioneered the heart team and shared decision-making for TAVRs. And I kind of wonder now that we're in the TMVR space, if we should have this discussion with our surgeons and have a shared decision-making with our surgeon before the first valve is implanted for a surgical valve prosthesis in anticipation of TMVR, since TMVR is no longer in its infancy. These are my disclosures. So my three basic goals are just to go over surgical valve design, which I found to be really wide, eye-opening, and then go through the basis of landmarks that we use for valve-in-valve implantation TMVR. And then the advanced discussion, deep dive, would be, should we have a multidisciplinary discussion before the first surgery, which I'm not sure our surgeons may like as much, but I really hope we can open the conversation moving forward. What we learned is that in 2019, this is a publication that was an expert consensus document by the cardiovascular surgery, the Europeans, Americans too, that basically surgical valve sizing and surgical valve design is kind of like the wild, wild west. And specifically in the sense that the labeling of surgical valves in this 2019 document, numbers 1 through 6, show that there is not a similarity or standardization of materials being used for surgical valves. The labeled valve sizing is actually not a valve sizing. There is numbers and there's inconsistencies between brands. The markings for our surgeons when they put a valve in, like which way is anterior, which way is septal, which way is lateral, is actually mixed depending on what brand they actually implant. The hemodynamic labeling is not standardized, nor is it conform tested. And there's basically no robust data. And the definitions for this and the problems have been here for decades. So I've been fortunate to be part of the CMC or Cardiovascular Masters Consortium for the past 2 years doing preclinical work, trying to really find comparisons. And we were able to do this study comparing 3 bioprosthetic mitral valves, specifically the Abbott Epic device, Medtronic Mosaic, and the Edwards Mitris valve, which just got FDA approval recently, in a head-to-head controlled study in porcine models. And what was really important to us was we wanted to kind of look at the 2019 documents put out by the surgeons and find out, well, what can we do to standardize this and help, because we have a real need in valve-in-valve TMVR. So this was a head-to-head comparison study. We wanted to look at the short-term acute durability, safety, and function of all 3 surgical mitral bioprostheses head-to-head. And this is not feasible in humans because if you think about it, we have so many anatomical variations in the human anatomy. Think about all of our TAVR population: septal hypertrophy, LV size, concomitant valvular heart disease, different blood pressures. And of course, we don't have necropsy or autopsy verification of the valves when we put them in. And all the valve-in-valve that we've done have been in diseased valves of some sort and different size and labeling. So we really can't do head-to-head. So we took these three surgical valves and did the controlled anatomical hemodynamic sizing. And we did it like we would do for a patient when we're doing TMVR implantation or surgical implantation. So all the surgeons were blinded to what mitral bioprosthesis they were going to implant until the time of surgery. All of our implanters were blinded to what type of surgical valve was actually implanted until the time of valve-in-valve. And then we did an epicardial echo, we did an intraprocedural TEE, and we did pre and post CTs, both at baseline after surgical valve was implanted, and then after a TMVR valve-in-valve was implanted, so we could have 2D and 3D data sets to compare outcomes and hemodynamics too. What we realized in talking to all of our surgical colleagues was that the current decision-making tree for surgical mitral bioprosthesis valve selection is kind of centered around three core subjects. One, the label size of the device, and that number that comes with the packaging. Number two is because the neo-LVOT data is very well understood now on the risk of valve-in-valve obstruction, the surgeons are adapting that to think about length of the surgical struts in the outflow to try to anticipate future risk of LVOT obstruction. And then the third thing that the surgeons consider is valve durability questions, kind of like type of leaflets and hemodynamic function. So what we did was we started with the bench testing, and we took three comparable valves, the Epic, Mitris, Mosaic, and they're all labeled 25 millimeters. And we kind of just measured them. And we saw was that on the far left, the Epic, which was labeled as 25, is actually only measuring like 19 or 20 on the internal dimensions from the leaflet edge to the leaflet edge as a transcatheter heart valve will be seen. On the middle, the Mitris 25 was around 23. On the far right, a Mosaic was measuring around 20. So a 25, depending on the vendor, does not reach 25, and nobody reached a true 25 labeling. And so we had to realize that what we thought was sizes was just numbers, and there was no standardization for it. When we looked at all the different sizes across vendors, like the smallest 25 device to 33, the variation was substantial. You can see that on the top left, the 33 Epic internal dimensions max and min was 28, even though it was labeled as 33. And then the Mitris was 29, Mosaic 28. What was even more confounding to us was if you flip the valve upside down and look at the ventricular edge, an Epic 33 valve is only 26 in the ventricular portion, which explains why we get a lot of constraints sometimes. And then on a Mosaic, it's actually uneven, 26 to 25. So numbers are really meaningless. And the question is, well, how does this impact valve-in-valve and the neo-LVOT? Well, if you think about when we did the first validation of neo-LVOT, on the far right is how we conceptualize the outflow tract. We conceptualized in green a virtual transcatheter heart valve implanted into a surgical valve. And we modeled everything, even now, according to all softwares, as a completely cylindrical round valve. And we anticipate that's going to land in the distal strut of the surgical valve. And that's where the concept of the strut length came. However, if our labeling and our valves don't actually get round, that actually impacts our neo-LVOT. And so this is the first part that we want to go into. The concept of the LVOT strut length in the surgical community and that it can cause outflow obstruction for valve-in-valve is something that it has been heavily misconstrued. So on the far left, you have a Mosaic valve, middle is Epic, and far right is a Mitris. And what we did was we had a 3D printed clear platform. We put all three surgical valves on there like a surgeon would have on a normal mitral annulus. And the concept would be bench measurements of the strut length would actually correspond to LVOT number of strut protrusion. It actually isn't actually true because on the bottom, when we turn the valves on the side, some of these valves, like the Mitris, actually has an anterior cuff that is raised in the atrial portion. So despite the fact that we can measure the strut length to be longer, it doesn't actually reflect how much left ventricle outflow tract strut is exposed in the LVOT. So it doesn't actually translate to what the risk for outflow obstruction actually is. The other concept that was concerning for us was that the strut sizes differ. So when we put a different perspective of these same three valves, the strut widths vary between the Mitris, Epic, and the Mosaic, with the Epic being the widest, but Mosaic being wide from top to bottom too. And this was a new concept to us because we know not everybody is going to be able to get a TMVR. Over 50% of patients have some kind of risk of anatomical outflow obstruction. So we resort to preemptive alcohol septal ablation, sometimes laceration of the anterior mitral leaflet. But the assumption in our predictive modeling and the assumption of these procedures is that while we're dealing with surgical valves that will have equal amount of neo-LVOT between their struts. And this is where when we took the image of all these valves, we realized that not everybody has the same amount of gap or true neo-LVOT between the surgical bioprosthesis because the strut widths are different. And the strut width is different between the anterior lateral and anterior septal struts that hug the LVOT, and the gap between those struts are going to vary. So some patients may not ever achieve a true neo-LVOT with lampoon prediction modeling because the surgical struts are going to be in our way. So smaller gaps would be smaller neo-LVOTs, which would be bad. And so it puts back more caution into our LVOT prediction modeling that we need to be more humble and cautious about what we do for neo-LVOT and not assume that every valve and valve is going to be okay and safe. So in looking at it this way, when I draw on the bottom left, the same arrow across all three valves and try to extrapolate them to their width, you can see that there is difference in the measurements between the width of each of these valves, thereby impacting the neo-LVOT for potential transcatheter valve and valve. What we also found was that I had alluded to the fact that when we do LVOT modeling, we assume we put a cylindrical valve into the surgical valve, and it's going to come out cylindrical. What confounded us was on the far left, each surgical valve is designed differently, and I'm not really understanding why just yet. So on the far left, you can see the EPIC device from the atrial cuff to the ventricular strut has actually a slope and cones in. The mitris is kind of columnar, and the mosaic on one side is actually coming in angled, and the other side is less oblique and less angled. So slope versus cone design actually is important and impacts our outcomes. If we think about it, what we did was we actually measured, well, how does this affect our outcomes? So we measured the surgical valve opening area, or how the leaflet tips actually open in these surgical valves, both by epicardial echo, and then we did it by CT again, so that we could do a multiplanar reformat so that we wouldn't be skewed by any two-dimensional images. And we found that there is some implication between the form of a surgical device and how much the leaflets can actually open. The EPIC with its slope design had a smaller surgical valve opening area than a mitris. On this top left EPIC, the opening valve area was like 1.89. And we know in mitral bioprosthesis, we consider it by MBAR criteria degenerative mitral valve failure if the area is 1.5 or less. This is a fresh out of the box surgical valve, and we're at 1.89, all under the same hemodynamic conditions. And so that's a little bit concerning because then it brings back to the question, are we really looking at these valve and valve outcomes in our registries the same exact way? And should we be comparing and consolidating all valve and valve outcomes to be the same? The best way to think about this would be like in the bottom left image, if you try to run resistance bands around your ankles, that's the best analogy I can give to potentially how these surgical stress might potentially be impacting leaflet function. So hemodynamics is what everybody swears by. If you can take a look at the EPIC valve mosaic mitris, the mitral valve peak gradient after implantation of comparable valve sizes was 9.2. And that corresponded with the smaller surgical valve opening area both by echo and CT, whereas the mitris peak to mean was 5 and 2.6 and corresponded with the surgical valve opening area that was much, much larger. So concepts of surgical valve form and function were something that really shook us up because we went into this thinking about we need standardization. We didn't realize how much standardization there actually needs to be in the surgical valve design. And on the far right, does this really matter and does it impact valve and valve? On the far right is basically a SAPIEN 3 balloon inflation. And what's most interesting about this when we inflate the balloon is we realize how delicate the structure of a stent is. It's just like chicken wire and chicken mesh. So a balloon can inflate the valve. But if I think about that balloon and that stent, it's never going to beat any kind of nitinol frame or any kind of metal frame in the surgical bioprosthesis. The surgical prosthesis is always going to win. The transcatheter heart valve is always going to conform. So we did the part 2, which was the mitral valve and valve. So we implanted 26 SAPIEN 3 and all 3 surgical valves. They're comparable baseline sizes and the same hemodynamic blood pressure, heart rate conditions, and everything. And what we found is, on CT on the far left, this is what we've seen in the valve and valve quite commonly commercially, but it never really had an aha moment until we started doing the analysis. Well, when the valves actually have a conal shape and have a waist, they actually have a smaller opening in the center of the valve. The same was true for the mosaic, and the mitras with a more columnar design had less waist and less skirt. And on necropsy, you can see the same deformation changes, that the valve actually was cinched at the atrial portion, thereby, you could get more pinwheeling and more leaflet touching. And on the far right, there's less. So then we measured it by CT, because we had the post-valve and valve CT. And we can see that the valve atrial opening dimensions of a 27 epic was only 21.8. And then if we went after post-valve and valve implantation, the peak gradient was 15.5. On a fresh surgical valve after TMVR valve and valve, that would be considered a failure by our current MVARC criteria. And you can see that the waist opening area, which is the narrowest part of this, was 329. We put a 26 sapien 3. 329 is like a 23 sapien 3 size. We never reached a 26 size. Now, the mitras was 414. None of these valves actually reach a true 26 sapien 3 size, but the amount of deformation was pretty incredible. But then there's also landmarks, and the importance of landmarks of valve and valve implantation. What we noticed was that surgical landmarks and fluoroscopic ability to see a landmark is extremely important. So in the epic, there is some visualization at the annular level, and then there's some, but there's nothing in the left ventricular portion. Mosaic has a circle around the distal end of strut. Mitras is fully opaque, and on a scale of 1 to 4, 4 being the toughest, our implanters definitely liked implanting in a more radial opaque environment because they could see and actually land the valve easier. But we had to scientifically prove that. So we actually measured on post-CT what extent of the valve for the transcatheter valve protruded beyond the surgical frame. And on the far left, you can see in a less fluoroscopically visible valve, we had more protrusion in the transcatheter heart valve beyond the distal strut of the surgical frame, especially in the areas around the left ventricular outflow tract, because you couldn't see. And the far right, in a more radial opaque valve, we have more reproducibility, and the valve actually landed closer to the frame of the surgical valve. And this is important because pre and post neo-LVOT modeling, we want to see the predictive modeling is actually accurate and useful. And we can see that the impact of fluoroscopic markers is very, very important. On the far left, you can see the protrusion when it's not a fluoroscopically visible valve. Far right, you can see the more consistency and less protrusion into the outflow tract, which affects the outflow tract too. And there was no change in outflow gradient. It was a very small study, but it was definitely concerning to see the differences by the actual fluoroscopic markers. So advanced techniques, and where I want to conclude is we have a lot of data to come out. And like you had mentioned, Paul, earlier, there's a lot of registry data, a lot of TMVR that we're learning about. And the data that shows the valve and valve outcomes right now says, does not actually say that the size of prosthesis actually impacts. We think that right now the size of prosthesis impacts the outcome, and we think the suboptimal expansion of transcatheter heart valves leads to frozen leaflets. And those are the reasons we have higher gradients after valve and valve compared to valve and ring. But none of this has actually explored the concept of surgical frame design, how that may be contributing to the gradients and the mechanisms of valve and valve failure, because we want to have better valve and valve outcomes for our patients. So our findings are definitely showing 3 key findings: that there is a correlation between the atrial opening and the waist opening and post-mitral peak and mean gradients. The frame dimensions definitely affect the degree of transcatheter heart valve expansion, and something that we know in practice, the ability to see fluoroscopically affects our LVOT outcomes and gradients too. So shape, radial opacity, internal frame sizing all directly impact the form and function. And we really need to do a deep dive of our registry data to really start accounting for this and these variations, because not all surgical prostheses function the same. Now, these are 3 patients that we've implanted valve and valve before. And I will used to look at these, I'd be talking about valve and valve and how I do neo-LVOT modeling. But now I look at these with a different critical eye and be like, on the far left, the anterior portion of the surgical frame of the transcatheter heart valve never fully extended. The posterior portion was longer. I can see the waist. Far right, I can see my waist at the atrial portion more too. So the hemodynamics and the biomechanical outcomes, we need to study more. And we're doing this in patients, but we deserve better outcomes. So really, in conclusion, from perception to science, labeling to standardization, surgical valve design needs a lot of help from imaging. The numbers are just numbers, and a lot of the charts are not really conducive to true clinical outcomes. And we need to do larger studies, and surgical valve designs and valve impact. We need larger studies to really evaluate this impact so we can help patients more. Because TMVR design right now is also following the path of surgical prosthesis design. If we think about different tendons, Medtronic Apollo devices, Sophia, everything else out there, same track, there's no standardization, 48, 52, 22. We don't know what the internal dimensions are, and hopefully we can do some regulation to help our patients more and more moving forward. Thank you.
Fantastic. So it was great to see you take a deep dive into really looking at these valves ex vivo and really understanding, and that's a different way of 3D printing. So
it literally is. It literally is. But it's a lot of fun to play with the surgical valves, and it comes into a lot of question like, have we really been studying this the right way? And hopefully we don't label valve and valve failures to be the same, and really look at it more like, how can we prolong it? Instead of just looking at the polymers and the leaflet types, we might need to relook at the original designs.
So my colleague Vinny Bapad, as you know, the creator of the Valve and Valve app, he has spent many nights and weekends cataloging a lot of the things that you describe and trying to figure out all those different standards of reporting, which are completely not standard, really.
Yeah.
When you think about it. But so I want to ask you a few questions about your presentation. I mean, first, what's Cardiovascular Masters? What is it? What is this group? Is this a foundation or a meeting or symposium? What's the Cardiovascular Masters thing? That the logo.
It's a consortium right now. It's Bill O'Neill, Brian O'Neill, myself, Richard Stack, Randy Chitwin, Tommy Karanasu. And we have a combined surgical background, imaging, interventional, and transcatheter and structural interventions with Richard Stack. And we try to do preclinical, and we want to really advance the field using a clinical approach to preclinical medicine. A lot of times in R&D and in clinical medicine, when we receive the device, it's a privilege to receive the device and have an R&D device to help our patients. And it's wonderful. But by that point, the device is already past design freeze, way beyond design freeze. So any ability to change the device and next iteration to help patients or get it into more patients has been negated. So for example, for TMVR, I mentioned that 50% of patients are excluded from clinical trials because of anatomic insuitability. And the question comes back to, are we designing devices for patients, or are we trying to find the right patient for the right device? And what we're hoping is to intercept in the preclinical early development phase of all these devices as startups, so that we can actually intercept and help more patients long run, so you could go from bench to bedside much quicker and have a more ideal device that actually serves patients instead of trying to find patients to serve devices.
So that's fascinating. So you're past design freeze, but you're pre-commercial is where you're at. And so you're kind of fine tuning perhaps some of the human factors, the market segments for the device, and making it more user friendly, I presume.
Yes.
I mean, that's a nice area to function in. And I have to say, in Minneapolis, we love working in that area too, because the EFS, like you said, the devices are human ready, but they could use a little bit of feedback, and that creativity makes it fun.
Yes. Yeah. And that's where, it's not, as an imager, I only know so much about imaging. The surgeons bring so much more wealth of information with other surgical expertise, what they see in the cardiac. So what all of our implanters have the catheter expertise and fluoroscopic expertise in the feel and tactile. So putting this group together as a true heart team approach to this allows the startup companies to actually build faster, and it decreases their time. People always say innovation from the first thought to the valve implanting the patients like 10 years. We should be better than that. I mean, our cars are designed faster than that too. So hoping that this approach using human terms, human anatomy, and our understanding will accelerate that for our patients.
Yeah, I mean, I've been waiting for TMVR for a long time. I mean, last week was the 7th anniversary of our first TMVR case, which was the first in the United States. It was April 8, 2015. I can't believe it's been 7 years since we did the first Tendyne here. I mean, it's been too long. So when you say 10 years, absolutely, it
Yeah,
takes way too long. And we look back and like you were involved with TAVR in the early days. I mean, there were patients dying who were waiting for TAVR, and
yeah,
patients were waiting here while patients in Europe were getting treated. It was really just, and I think a lot of that helped lead the FDA to help change things too, to where we had more access to EFS than ever before because
yes.
they realized we had patients who were waiting.
Absolutely. The hardest part in the innovation sector and disrupting, as you've been a part of so much, is telling the patient who you want to help that, I'm sorry, we don't have a size for you. And hearing the family say, well, why don't you have a size for you? And it's like, we don't have that in development. It's not available. So being able to identify this hopefully will help. It's not going to be a cure-all, but hopefully it will help align the market needs more and expand the populations more. And I think that's really thrilling. And the impact of that is going to be really, really big.
So I want to go back to your presentation that you had earlier and talk a little bit about some of the human application there. So, looking at those different valves, there's certainly different struts and different widths and such. Did your paper simply I'm just going to ask this, did it simply just validate that we should be using the MITRIS valve instead of the EPIC? I mean, it seemed like such a better valve compared to the Mosaic or the EPIC.
Well, it did not reach statistical significance. It was an early feasibility study, so it was only 12 clinical animals. So from that aspect, what it definitely showed was there's a definite clear difference between form and function between different types of surgical bioprosthesis. And it opens a lot of scientific opportunity, because one of the things we have to really look at is the valve outcomes in our TVT registries have not been defined by type of surgical valve. And people always, the reason hot stuff in the literature is, well, what mitral mean gradient is okay after edge-to-edge repair? And the same question is asked for TMVR. What mitral gradient is okay after a valve-in-valve? We started with a mitral valve gradient of 12. Now we're down to mitral valve-in-valve gradient of 8. Is that a good outcome? Or how do we actually assess that? And hopefully this will allow more scientific studies that are larger to actually really study these valves and get a consensus about, okay, which one is better? Is there a better valve, and is there a better design for our patients? A lot of questions remain unanswered. I think this validates that we need a more urgent call for standardization. And if the surgical community wasn't able to do it, maybe from the transcatheter side, KOLs like yourself can help push that effort from structural and say, we need more because we're doing valve and valves and we need better outcomes.
Yeah, so when I see that I have a mitral epic or BioCore that needs a valve and valve, I cringe, because I just know it's going to be hard to see.
I cringe too, because
I take
I pay more attention on TE.
Yeah, that's true. I need to rely on TE more, but I get a lot of extra radiation exposure when that happens. Tell me about how your results inform Lampoon and when to do Lampoon.
So preemptive alcohol septal ablation and Lampoon are two of the methodologies, and then RF ablation is starting right now too, as a possibility too. And laceration of the anteromitral leaflet in valve and valve is feasible, but the one thing that we found about this is we had to really be careful of the calculations we use for neo-LVOT modeling. We assumed that everybody had the same opening area on every single surgical valve design, and that the skirt neo-LVOT, or what we call about when we lift a valve more atrial, is going to be the same just because we lifted more atrial. Then it's not going to be the case across all surgical valves because of the design. And so how is it going to affect our calculations? We might not be able to predict it, and we just have to be able to realize that and account for that and understand that risk. Preemptive alcohol septal ablation is another technique where we do the septal perforators, but the limitation is not every septal perforator is the same size. So and it may or may not run the area we need it to, and we may not get the same outcomes. But having these tools and these available tools of RF ablation, alcohol septal ablation, myectomies, Lampoon, are all going to be important to move forward until we get to a more ideal design for these valves.
Yeah. Well, I think it's it's amazing because, historically, when if you look at like what a surgeon does, they never looked at the CT before they went in and replaced the valve because they're just going to cut out that leaflet or give it a few slices. And so but now, I mean, I have to have a CT. I mean, you described in the beginning how you kind of eyeballed it with 3D printing and such, but now it's malpractice to think about mitral valve and valve or TMVR without a CT. You just can't, right? I mean, do you see a future where you can do it all with just TE? I mean, tell me about that, about sizing and placement and such. I mean, it would be nice if we didn't have to always just rely on CT, if we could transition more to intra-op echo.
Yeah, I wish. That's a wish. But the problem just is that alternative access pathways, the TE probe can only show so much of a sector size. So I can't see the legs. I can't do sentinel evaluation, cerebral protection evaluation. So I think an emergency TE is going to be okay, but it won't be able to do neo-LVOT prediction modeling to the accuracy that we need just because the frame size and the frame rate is not good enough. I hope that CT will become more robust. It's fast. It's really fast. And that the slice thickness is so small at 1.25 millimeters. We can see everything from the eyeballs down to the legs. But the question would be, do we need TEE in the future? I do see TEE being replaced by good 4D ICE if the 4D ICE becomes better technology. But it will be a lot of development in the next 5 or 10 years, and it'll be fun to watch what comes into play as the market grows too.
Yeah. Oh, let's touch this in the closing minutes. Let's touch a little bit about on ICE and such. So are you doing the ICE at your hospital, or is the interventionalist doing ICE?
So we just started doing ICE. There's been limitations to getting access to ICE. We're looking at 4D ICE availability with all the supply chain issues in the pandemic. Not everybody has enough ICE catheters. My vision is to be holding the 4D ICE probe and doing the ICE. Before all of this, before I got interested in ICE, I used to say that ICE stood for I can't see. Because the TE technology was way better than the ICE, but I definitely see the 4D ICE technology is coming and getting more robust. But the design platforms are a little bit cumbersome right now because now I'll have to bring in a sonographer with me. I'm going to do the ICE because I can't unsterilize my hand on one probe and then actually touch the machine 6 feet away. So a lot of progress that can be made in this future development. There's some cool dielectric imaging technologies coming out too that might replace ICE. So a lot of development in this field that I am hoping will advance what we do beyond just a TEE probe.
I mean, I'm working on learning ICE as well. I mean, I can do ICE for PFO and ASD. I mean, that's easy. That's pretty straightforward. But all the other imaging to try to get to be like what you see with TE, I look at the control panel, and it's kind of like what you would see when you go into an airplane and look at the pilot box.
Yeah.
See all these switches that just don't mean anything.
Yeah.
And
exactly.
so to me, because I'm just lost. And so if you want to know how I feel when I look at those panels, switches, and controls that you're trying to get to unsterilized, I just don't even know which ones to touch. So one of these days, I'm going to have you and some others come in and show me which ones are the ones to touch.
No, I'm not that good with new technology. When they show me, oh, you can swipe screen here, I'm like, I don't want to swipe. I just want to touch. That's how I feel and move on.
Well, that'd be amazing to be able to control things like, by motion and such.
Yeah.
No, but I'm just going to check the chat here, make sure there aren't any questions in the Q&A. Oh, so there's a question about early career imaging physicians. I think we talked a lot about that, about how to be competitive. So, but it really has been just a great pleasure, Dee Dee, to have you here. And, I hope to have you more at CVI in the future and get more involved with us because I think where you sit in that continuum of new technology and innovation on the cusp of bringing it to the larger patient population out there, it's a really unusual, unique position that only a few have. in this world. And it's great to hear your perspectives on that.
Well, thank you so much, Paul. It is truly a privilege to work with you in the Minneapolis Heart Foundation, and we'll be privileged to work more with CVI. For this question, for all the people out there, there's no competitiveness in interventional imaging. We could use all the help we can get. We have a fellowship, so hey, we're always looking for fellows to join us. We have spots open for 2023, 2024. Just give us a holler. And there's plenty of sharing opportunities, and we'll make you grow and elevate you. But Paul, it's always a privilege. Thank you so much for this honor, and thank you for all your support to the interventional imaging community. Moments like these and your support is just priceless. And we really cannot say how important it is. And we can only feel it and tell you that thanks for making us feel validated and real. Much appreciated.
I owe so much to you guys, because I can't do anything. You can't treat what you can't see. And you guys always show me the way. So thank you, Dee Dee. And with that, I'll close this webinar and thank all of you for joining us tonight. And I hope you all make it to CVI in Denver. And please feel free to contact us if you have any questions. Thank you very much.